Hello, good afternoon, everyone.
Good afternoon.
I hope everyone is doing well.
By His grace.
Yes, sir. Thank you.
Yes, I'm doing well.
Thank God.
Thank God.
So my name is Kwajo Safumafo. I'm the lecturer for microbiology for this semester. I believe this is midwifery class 300.
Yes, sir.
Yes,
Yes, sir.
All right. So let me share my screen. Please, can someone confirm the full screen?
Yes, sir, we can see.
All right.
we can.
So at this point, I would plead that you mute when you have a question or a comment, forth your question or your comment. So these are the course objectives. At the end of this lecture, or for the microbiology course in particular, you should be able to give a brief history of microbiology and then describe the various ways microorganisms are identified. You should be able to describe the routes of entry of these microorganisms when it comes to causing diseases, and then describe the ways of preventing infections. Then you should be able to apply this knowledge in your practice as a midwife. The course content is displayed and we shall tackle all of them. So we will first start with the history of medical microbiology, the habitat, and the conditions that are necessary for microbial growth, which is the unit 1. This is the continuation of the course content. So history of medical microbiology, the habitat and conditions suitable for microbial growth. I'm sure you've heard of microorganisms or microbes or germs. So micro means small or tiny. Bios means life. Logos means to study. So microbiology, the study of small life. Human beings are the bigger lives, or the macro biology aspect that we study. But these ones, we don't see them with our naked eyes. So you need to use an instrument called the microscope to magnify these microorganisms or these tiny, deals with the study of these small, tiny, minute organisms and their effects on humans. So for the sake of your, there are other beneficial effects that we get from microorganisms. And in the course of the lecture, we'll be talking about some of these benefits, but we'll hammer more on the diseases. So these microorganisms can appear as single cells. So when you take the human being, for instance, you have so many cells. That come together to make a tissue. an organ. But these microorganisms, because they are very small and simple in form, they have single cells. So we have unicellular microorganisms and multicellular microorganisms. They are multicellular, they have many cells, but they are still small, very tiny, that you need the microscope to see. So microorganisms are of various. Hello? Someone has a question?
The line is breaking. The line is
Oh,
breaking.
all right. Is it better now?
Yes, sir.
Yes, sir.
All right. So let me take that again. These microorganisms appear as single cells or multiple cells. So we have unicellular and multicellular microorganisms. Those that are unicellular are typically the ones that have very simple forms. The multicellular are the ones that have specialized functions in addition to what they do. So they have competitive advantage over the unicellular organisms, because the more specialized cells you have, the more diversified your functions become. So microorganisms do not include viruses. Take note, a virus is not an organism, because an organism must have a cell. The cell is the basic unit of life, and viruses do not have cells, but they can reproduce or replicate. They can multiply. For a virus to multiply, it needs to hijack or get access to a living organism cell before it can begin to multiply or replicate. So take note that a virus is not a living organism, but a virus is very, very tiny. You cannot see the virus with your naked eye. So you need a microscope to see these viruses. Again, viruses cause infections, just like some of these microorganisms. That is why we include viruses under the study of microorganisms. So one, they are not living organisms, but because of the diseases they cause and the techniques that we apply to study these viruses, which are the same as what we use to study the microorganisms, that is why we include viruses under the study of these viruses. Please, is the line okay? Can someone confirm now?
Yes, sir.
Yes, sir.
It's okay, it's clever.
Okay, so yes, thank you very much, Martha. So we look at the history of microbiology. And this is going to be a brief history because this is not a history class, this is midwifery class. So I need not bother you with so many information when it comes to the history. But there are some key names that you should be familiarized with when it comes to the field of medical microbiology. So for many years, people were wondering where diseases were coming from and how they were spreading from each other or in the community. And it took several years with several theories and beliefs for people to explain how diseases come about. And as we know, seeing is believing. So if there's nothing to see, why do you tell me that these are little animals? It was hard for people to believe that. So several beliefs or theories were proposed. Some thought that diseases were caused by demons, which of course some still believe. Witchcraft, bad luck, the wrath of God. So if you do something wrong, then God will invoke disease on you. Curses in the family or individuals, evil spirits will visit the community and then come along with diseases. So these were the thoughts or the beliefs they had. So there were several theories, and some of these include the supernatural theory, beyond the natural. So it has to do with divine or godly processes that will bring about the diseases. Then we have the Hippocratic or the miasma theory, the theory of contagion, contact between persons. Then the germ theory. Then the theory of epidemiological triad. Then multifactorial causation theory. So the disease for the epidemiological triad, you have the host, then the pathogen, and then the environment. And you can have disease caused by multiple factors. So all these beliefs were put together, and then we understood the microorganisms, and then microbiology evolved with time. So supernatural or divine forces were thought to be the cause of diseases. And at that time, they attributed these to demons, ghosts, evil spirits. And so magicians were diagnosing and treating. So we had witch doctors, priests serving as healers, because they had the eyes to see what is beyond what the human eye can see. So they use magic spells, exorcism, to drive out evil spirits from the community, or producing chemicals or drugs that will make someone nauseate or bring out some things from the body. So they were using all these means to cure or treat. And some way, somehow, it was working. So people believed. People still have these beliefs anyway. Then we have the Hippocratic or the miasma theory. So Hippocrates, as far back as 460 BC, came up with this idea that was contrasting what the supernatural theory was proposing. So he believed that diseases were not brought by divine or supernatural powers, but because of the four humors or the bodily fluids in humans or animals. So he focused mostly on the humans. So blood, yellow bile, black bile, and phlegm determine the health and the temperament or physical attributes of a person. So if we see that somebody is producing a lot of phlegm, then we suspect the person has some infection. That was the belief of the Hippocratics. So example, they believe that if someone had epilepsy and the seizures appeared because of the foamy substance that were coming out, they suspected that producing phlegm was the cause of the disease. So this was not by divine or supernatural force. So they established a relationship between weather, the atmosphere, and disease. And because of this, poisonous vapors or miasmas were responsible for these diseases. So when you have decomposing materials, swarms in areas, they could give rise to diseases, and not evil spirits that are visiting with these diseases or cases from gauze. So it became a dominant issue. Europe, China, India, they were all buying into this belief because there were several outbreaks: cholera, chlamydia, the bubonic plague, the Black Death, that resulted in the death of so many people, millions of people. So they thought that these were caused by bad air in the communities. So others also thought that it's not only the bad air, but there are some little animals that we cannot see. There are some worms, there are some seeds that are within these poisonous substances or these air particles that are responsible for the disease, and not just the bad air. So there was an evolution of these Hippocratic theories. So this led to the theory of contagion. So Hieronymus Fracastorius also said that disease is caused by contact between persons, and that it could be direct, so from one person to the other, or droplets, so somebody sneezes or coughs, then these particles or these droplets would move from one person to the other, and then the person gets the infection. Or we have fomites or fomites, depending on how you pronounce it. But these are contaminated materials that have the infectious agent. So he said that disease is contagious and quarantine is the answer. So disease can spread from person to person through contact, droplet infection, or fomites, and quarantine is the answer. So let me ask, what's the difference between isolation and quarantine? You are health workers. Can someone tell us difference between isolation and quarantine?
Isolation is when you separate a person having the disease from uninfected people. Whilst with the quarantine, we quarantine them for the period of which the person shows the signs and symptoms of the disease.
That is right. Thank you. So before the identification of these microorganisms, they suspected their presence, but they could not prove it. They didn't have the instruments to actually let people realize that microorganisms were present. So as far back as 55 BC, Lucretius and Girolamo Fracastoro were saying that these were caused by living creatures. So others did not believe in the supernatural theory or the miasma or Hippocratic theory, but they believe in spontaneous generation. So out of a non-living material, life could arise. This was the belief of some of these people. And Aristotle was one of the champion advocates of these theories. Please mute. So he described that the kinds of species could be generated from various substances. So life comes from a non-living material. So seeing maggots in decaying matter, your grains fermenting, wounds getting worse, gave rise to the belief that microorganisms arise from decomposing non-living matter. So you could see maggot from the rotten meat or mushroom from rotten wood. So this made them believe that spontaneous generation or abiogenesis is possible. So you have moisture in the atmosphere, then combined with slime or manure, then you wake up, you see fireflies, worms, bees, wasps, then soil giving rise to mice spontaneously. This was their belief, but they didn't know that there were some factors that were responsible for attracting these living creatures. So mice, they live in dark areas and untidy areas. So once you provide the food they need and these conditions are favorable, they could come. It's not spontaneous. So Francesco Redi performed an experiment, but later on, John Needham said that once you boil your meat and then you have the broth, which is the liquid part, you seal it, it can still have growth, so it's spontaneous. So we'll look at some of the experiments that were performed by those who disproved the spontaneous generation theory. So they perform experiments to conclude that life comes not from non-living materials. So Francesco Redi said that maggot is unable to grow on meat once you cover with gauze or you cover with a cork. And Schwann, Frederick, and then von Dosch also allowed air to enter the flask. But once you heat, it becomes sterile. So heating eliminates or kills these microorganisms. Once you prevent the vectors from getting access to the meat, they are not able to lay their eggs, and then you don't have contamination or maggots coming out. So dust also contains microorganisms. So once you take care of the dust, you omit or get rid of these microorganisms. But there could be some resistant forms called the endospores that could still be in the dust or the environment, even if you apply tinder. So Lazzaro Spallanzani said that there is no growth if you boil and then you seal the flask. But again, there are some organisms that are anaerobic. They be able to grow very well. So contamination from absence of air is possible, according to Felix Pusher. So they performed several experiments to disprove what the other side believed about spontaneous generation of life or microorganisms. So this is the experiment that Francesco Redi performed. So he put meat in a flask, uncovered, and allowed flies and air to settle on it. Then after some time, formation of maggot was observed. So this was not spontaneous. It's because of the exposure of the meat to the flies and then the air. Then he put another meat in a container, by this time sealed. But because the flies could not get access to the meat and microorganisms were prevented from having contact with the meat, it stayed sterile. So this was not spontaneous. Then another member said that air, which is fresh, is needed for the growth of microorganisms. So he put another meat in the flask, and this time covered with gauze. But because they didn't have access to the meat, the flies didn't have access to the meat, maggots could not form. So this disproved the spontaneous generation theory. Someone has a question? All right, let's move on. So the field of microbiology became dominant when Robert Hooke invented the microscope, and he was able to document what he saw, and said that living organisms contain little boxes or honeycombs, something that look like honeycombs, called cells or prisons. So that's how come we got the name cell. So every living organism is made up of a cell. Then use this simple magnifying lens to observe the plants and then some leaves and some living materials to confirm that when you take any living organism, it's made up of these small boxes called cells. So he suggested that all living things are made up of cells. Then another great scientist, Antonio van Leeuwenhoek, also developed this single hand lens that could be used to observe living organisms. So he is known as the father of bacteriology and protozoology, so the father of microbiology, because he was able to use a simple lens to describe living materials or living organisms. But this was a simple instrument, and at that time there was no electricity, so you need to be patient and then have it read in the presence of sunlight. Then he documented all these things, and this made a breakthrough in the field of microbiology. So whatever you see in the world, in the hospitals, please document. It may be your starting point for breakthrough. Then we move to the germ theory or the biogenesis, which was finally done by Louis Pasteur to disprove the spontaneous generation theory, because there was a debate ongoing at that time. So he introduced what we call pasteurization, eating intermittently to shock or inactivate microorganisms so that your food or your wine does not go bad. So this led to the germ theory. He says that cells can only arise from pre-existing cells and not spontaneously. So he is credited for his works in the field of bacteriology. Then later on, Robert Koch also performed experiments, which we'll look at in the subsequent slides, to prove the germ theory that Louis Pasteur had proposed. These were also supported by other scientists, including Rudolf Virchow, who was able to study how cells multiply. So binary fission was credited to Rudolf Virchow. So this simple experiment performed by Louis Pasteur, at that time, the wine in France was very expensive, and within a short time, microorganisms would spoil it, so they were losing a lot of money. So he was able to save them a lot of money with this simple experiment. So once you apply heat to your wine or to your food, microorganisms are not able to withstand, so it stays sterile. But if you break the neck of this flask, because of the exposure of these microorganisms to your wine or your food, it goes bad. So growth will occur, growth of microorganisms will occur. Again, if you don't break the neck, it stays intact. So he was able to save their wine by application of heat, so pasteurization, and then covering of the food products right after production. So once you apply heat, you're able to make the substance sterile and there is no growth. But once you open it, there is growth. So this simple experiment disproved the spontaneous generation theory. So this is called the swan neck flask experiment. This is a swan neck flask. So Louis Pasteur proposed the principle of fermentation because at that time they were making money from wine production. And he also introduced pasteurization of milk to save the dairy industry. And sterilization techniques to kill all forms of microorganisms, including their spores. Then the germ theory of disease. So if microorganisms can make food or wine go bad, when they get access to the human being or the body can also become sick. So microorganisms make wine sick and they also make human beings sick. So this was the analogy Louis Pasteur proposed, and he was right to that extent. So he was able to save the wine in France, and we still apply some of his techniques currently or in modern practice. So he said that good wine contains yeast and the bad wine contains bacteria. So you need to inactivate the bacteria so that it doesn't go bad, because the bacteria will produce acetic acid and then it will make the wine taste funny. That's why I introduced pasteurization to inactivate bacteria that may be present in the wine or milk. So all the diseases that were associated with milk, wine were controlled due to what he was able to do. Then he proposed that microorganisms are responsible for infectious diseases. Not all forms of disease, but the infectious types, the ones that are caused by microorganisms. So the germ theory of disease states that many diseases, not all, are caused by the presence and action of these microorganisms within the body. So before disease can occur, these organisms must have access to the body. And gradually it became accepted and then ruled out the miasma and then the spontaneous generation theory in Europe and then other parts of the world. And to date, we rely on the germ theory to control infections. So as time went by, development of aseptic techniques were developed, or these came into being. And these are strict adherence to practices that will minimize contamination within the facility. Hello. Please mute. Okay, someone is asking to go over the diagram. I hope this is the diagram you are talking about. Can someone confirm? The first one, this one.
No, the meat.
The one by Francesco Reddy. This one.
Yes, sir. Yes.
All right. So he wanted to disprove the spontaneous generation theory, so that life is not coming out of non-living matter. So this meat is non-living. And according to them, life was coming out from non-living matter like this meat or tables. So these are the experiments he performed. He set up 3 flasks, one with sterile meat, but left open. Then allowed air to be in contact with the meat. So flies could easily step or settle on the meat to lay their eggs, and after some time they saw maggots coming out from this meat. So those who were in favor of the spontaneous generation thought that this meat, which is non-living, had given rise to living materials, the maggots, which will eventually become flies. Then he told them that this is not possible because life comes from a living organism or living matter. So he put another meat in a flask and then covered it. And this time, flies could not have access to the meat to lay their eggs. And so no maggot was seen. So if no maggot is seen, then life does not come from non-living matter. It must come from a pre-existing living material. So this simple experiment was able to disprove the spontaneous generation theory. Then they came back to say that you need fresh air for life to continue. So he said, okay, this time I'll not use the cork. I'll use gauze so that the fresh air you are talking of can have contact with the meat. But the gauze did not allow the flies to settle on the meat to lay their eggs. So this made them see that no maggot was growing on the meat, to disprove the spontaneous generation theory. So this led to the theory of biogenesis, cells arising from pre-existing materials and not spontaneously, not coming from non-living material. I hope it's clear. Hello.
Hi.
Hi.
Yes.
Hi.
Hi, sir.
The one who
Hi.
wanted clarification. Are you okay? Right, thank you.
Oh,
So move on.
di mama nyona pan yuba ce yi. A fin ga na yi ce sama na zon barka. Ah,
Please mute.
ne ka
Please
dai
mute.
fin na kan da mu an ce ba sama na zon baro. Awla ma haba na yi kokori.
Please mute. Priscilla Broker, please mute. Thank you. So aseptic techniques came into light, and this helped physicians, the health workers, and the scientists to reduce hospital-related cases, very contagious. It led to so many lives lost because the physicians were not adhering to some of these practices, and they were cross-contaminating the patients. They were also contaminating themselves or infecting themselves with these microorganisms. So once the development of aseptic techniques came about, it saved them from these harmful microorganisms. So Joseph Lister is also credited for the introduction of these antiseptics. So he used chemicals to control microorganisms in the health facilities.
Oh, yeah, so they are
So they use... Any question, please? Please mute. All right, thank you. So they used chemicals instead of heat to control microorganisms. So carbolic acid or phenol sprayed in the air around the surgical incision or the wound was able to control microorganisms and prevent the spread from patient to patient. And these reduce the number of post-operative infections at the hospitals or facilities. Then he introduced the mouthwash, which we named after him, the Listerine mouthwash, to also help control infections in the mouth. And these days, we still continue to use antiseptics. So an antiseptic is a chemical that is used safely on a living tissue to control microbial growth. So Robert Koch also contributed to the field of microbiology by developing pure cultures. So he was able to isolate the organisms that they are saying is invisible and grow them using favorable conditions. So these favorable conditions would make the organisms multiply. Remember, I said that microorganisms are very small. You cannot see them in the naked eye. But once they multiply and they become billions, you see them as colonies on the petri dish. Then you can isolate this microorganism. So this is what Robert Koch was able to do using potato slices, which contains nutrients. And then later on, they cultured these microorganisms. And he was able to prove the germ theory that Louis Pasteur and the others had proposed. So he worked on so many organisms that cause infections or diseases like tuberculosis, cholera, diphtheria, typhoid fever, gonorrhea, pneumonia. Because this time, patients came in and he was able to take samples from them and culture the bacteria that is responsible for the disease so that they could treat accordingly. So he did very well in the field of microbiology. So this experiment Robert Koch performed proved that disease is caused by microorganisms or germs. So at that time, people were having pneumonia. So he took sample from those who had been infected and then injected into a mouse. The mouse died. Then he took sample from this mouse, which had died, and put on a medium. So when you say medium, we talk of nutrients that support growth of microorganism. So he incubated under favorable conditions, and after some time, he saw growth of these microorganisms on the petri dish that contains the medium. So this time we call it culture. So when you say culture, culture is a medium that contains the growing microorganisms. Then he took some of these colonies on the petri dish or on the medium and observed under the microscope. So later on, we'll talk about staining. So he prepared a slide and then stained and then observed under the microscope because he wanted to see their morphologies. So he recorded what he saw, and then he took some of the colonies from the petri dish, not the dead mouse, but this time from the petri dish that had grown the microorganisms. Then he took a healthy mouse and injected the microorganisms from the petri dish he had cultured into this healthy mouse, and the mouse died. So the disease is reproduced in the second animal. So there is contact, and then the organism dies because the same organism is produced and then the effect is seen. So we wanted to see if the organism that made this healthy mouse die is the same as the one he isolated here. So he took samples again from this healthy mouse that had died and then cultured on a medium. incubated for some time, and then observed under the microscope. Then he saw that indeed it was the same as what he had isolated from the first mouse that had died. So he was able to say that germs cause diseases. And for every disease, you should be able to isolate the organism that is responsible. And once you introduce into another organism, it should be able to express the same characteristics. So this led to the Koch's postulate that says that microorganisms must be present in every case of the disease. This is what Robert Koch said at that time. Secondly, he said that these microorganisms that are present in the infected organism must be isolated and then grown in pure culture. If it is there, then you must be able to isolate it and grow. This is what Robert Koch said. Then there are specific diseases that must be reproduced, or characteristics that must be reproduced, the symptoms from a pure culture when you re-inject into a healthy, susceptible host. So the first condition that you observed when you introduce this organism from the pure culture into a living, healthy host that is susceptible, it should be able to exhibit the same characteristics. So if the symptoms are coughing and headache, you should be able to see the exact symptoms or characteristics or signs. Then he said that microorganisms must be recovered once again from the experimentally inoculated host. So the one that was healthy, that you injected the microorganisms into, after exhibiting the characteristics, you should isolate the same. Hello? Any question, please? All right, let's continue. So you should be able to isolate the same organism as you saw in the first from the host that was healthy and had been injected with the organism to exhibit the symptoms or the characteristics. But not everything that Robert Koch said was true. There were some modifications to be made. So let's look at the exceptions to what Robert Koch said, although some were true. Yes, Esther, you have a question. Please go ahead.
Sir, please. The experiment, the first mouse, I didn't get it. Was the first mouse healthy as the second one, or it was sick?
So he was trying to establish causation of a disease. And at that time, there were some organisms that were causing deaths. So he had, in his lab, saved some of these microorganisms, these bacteria. So he had these lab rats or lab mice that they were using for experiments. So he just went to his fridge, took some of the microorganisms that he had saved from previous patients, and then injected into the mouse, this first mouse, to see the effect. And it died as the other patient had suffered. Then he isolated this organism that was responsible for the death of the mouse by taking a sample, culturing on a medium, and then observing under the microscope. Then taking some of these microorganisms from the petri dish to inject into another healthy mouse to see the effect. And then it also died. So Esther, please, is it clear?
So, sir, please. The experiment was like the human being is sick. He took the microorganisms from the human being, cultured it, and he gave it to the lab mouse.
Exactly.
Then
Exactly.
afterwards, he took it back again and into the second one.
Exactly. So this was a lab experiment.
Okay, thank you.
So it's like we have malaria in our case. So somebody comes to the hospital, you take blood sample from the person, you do the lab test, it's positive, plasmodium. Then you can take some of these plasmodium that are compatible with other animals and then inject into them to see if they will develop the symptoms or signs of malaria. So this is what he did with bacteria into this mouse. All right, thank you. So exceptions to the Koch's postulate. Let's look at what he said earlier. Microorganisms must be present in every case of the disease, and they must be isolated from the disease host and grown in pure culture. But the exception is that some microorganisms that cause diseases are unable to be cultured on artificial media. So the disease is there, all right, but it's difficult to grow that organism on artificial media. It only grows in the host. So this was an exception to Koch's postulate. Example is Treponema pallidum, the organism that causes syphilis. You can take the sample from the patient, you can do the test to confirm, but growing the Treponema pallidum on artificial medium is very difficult. Research is still ongoing to come up with media that will support the growth. So this one was an exception to Koch's postulate. There are some infectious diseases, but it's not always the case that you can grow the organism that is responsible. Then the other exception is some diseases are caused by multiple microorganisms. So you could have mixed infection, or you could have fever or diarrhea. So diarrhea could be caused by cholera. Diarrhea could be caused by Salmonella typhi. Could be caused by Campylobacter. So this was an exception to Koch's postulate. If patient A complains of diarrhea and vomiting, you take sample, you isolate, and it is Vibrio cholera. Patient B presents with diarrhea and then vomiting. You take sample, you culture, and it's not Vibrio cholera, but it's Campylobacter. So there's an exception to Koch's postulate. It's not every disease that you isolate the same organism. There could be different organisms that are responsible for this. Then we have some organisms that cause several diseases. So you could have bacteria that is causing meningitis. The same bacteria is causing cardiovascular infections or disease. The same bacteria is causing blood-borne infections. So this is an exception to Koch's postulate. So you can have urinary tract infection caused by bacteria. That same bacteria could cause gastrointestinal tract infection. And some diseases have variable signs and symptoms between patients. Patient A has malaria, presents with headache, fever, and then vomiting. Patient B also has malaria, presents with headache, fever, no vomiting. So this is an exception to Koch's postulate. And because you want to conduct experiment does not give you the right to use human beings for experiment. There are some infections that are only produced in human beings. Human beings are the only host. So you cannot use the rat, you cannot use the mouse. And because of that, you are not forced to use a human being. is against ethics. So these are exceptions to Koch's postulate. Then further on, Schleiden and Schwann proposed the cell theory based on the observations made by the other pioneers in microbiology. And these were modernized to become the modern cell theory. So all organisms are made up of cells, which is true. When you see an organism, it must be composed of cells or a cell. The new cells are always produced from pre-existing cells. So the pre-existing cells will reproduce or undergo multiplication to produce the new cells or the next generation or the daughter cells. And the cell is the structural and functional unit of all living things. The basic. Beyond the cell, there is no basic. The activities of life cannot go on if you eliminate the cell. So all living activities go on within the cell. Then cells contain hereditary information or genetic information that is passed from one generation to the other during cell division. So the traits of the parent is inherited by the offspring. And this is not spontaneous generation. So there must be a pre-existing cell passing this trait in the form of genes or the DNA that has this information to express the trait. Then all cells are basically the same in composition when we come to the chemical composition. So carbon, nitrogen, oxygen, all cells are basically the same in composition chemically. And the metabolic activities are also basically the same because they have the same chemical compositions. So these are the modern cell theories that we still hold on to. The history of microbiology and discovery of microorganisms and the techniques go on and on and on. Research is still ongoing, so it's non-exhaustive. I'll not bother you with the history. So when it comes to vaccination, we talk of Jenner. People have demonstrated a lot, so they are credited for that. It goes on and on. Discovery of the first antibiotic by Alexander Fleming, penicillin. Now we have vaccines. We have insulin that can be produced using microorganisms. So genetic combinations or recombinant engineering is also possible. It goes on and on and on and on. Sequencing of the organisms, having to know the chemical composition, the genetic material, how the DNA appears or the nucleotides appears in these organisms. So we have come far in the field of microbiology. Now, what is there for us in the future? We have emerging infectious diseases, and even the old ones are becoming widespread and very, very destructive. So we need to also devise strategies to control them. COVID-19 came, but before that we had the SARS. So we have reemergence of these diseases, and new ones will keep coming because of the continuous exposure we have with the reservoirs. So we must find ways to stop these by chemotherapy or by other means, preventive measures that will make us stay safe. We also need to devise strategies to diagnose them properly, because if you miss the diagnosis, you also miss the treatment. In the hospitals, we also have to practice aseptic techniques so that we don't encourage nosocomial infections. We need to continue to do research to ascertain the association between these infectious agents and the chronic diseases. So those with autoimmune diseases, how do we help to control these? Then microorganisms are evolving all the time. They are changing. So we also need to study how they are changing and then be ahead of that to control them. The microbial communities, they live in biofilms. So once we eliminate these biofilms, we also having access to good quality life. And we can also engineer some of these products from the microorganisms to help in medicine. So we come to the second and third topics under the Unit 1, which is habitats and conditions suitable for microbial growth. But before that, does anyone have a question on the history, the experiments that they performed? All right, so we move on. So microorganisms are ubiquitous. They live everywhere. They are present everywhere that you can think of. So everywhere you go, you find microorganisms. Even places that you cannot go, microorganisms live there. And they form the largest biomass below and above the earth. Hello, please mute. So they can survive very high temperatures, very, very low temperatures. In the fridge, in the freezer, microorganisms will survive. Volcanic eruptions, microorganisms, some of them can survive. But the good thing is, the ones that cause diseases, they don't survive these extreme temperatures. So your body is a perfect condition for them to live. They can live on food, they can live on petroleum products, toxic waste that are harmful to humans, microorganisms, some of them can live in them. That is why you take drugs or give drugs to your clients and sometimes it doesn't work because they can become resistant with time. So there are typically around 40 billion bacterial cells when you take 1 gram of soil. So you can just imagine the size of the gram of soil and then the number of microorganisms that you can find in there. They are tiny. If you take 1 milliliter of fresh water, you have millions of these microorganisms. So if a patient coughs in the ward and you have droplet infection, you can imagine the number of microorganisms that have come out to infect the other patients or you, the health worker. That is why you need to protect yourself when you are in such facilities. On rocks, in soils, everywhere, there are microorganisms. In your shower curtains, they are there. Your curtain board, they are there. Under your carpet, anywhere in the atmosphere, they are there. So once you open your mouth to talk, they are entering. But your body has some barriers to prevent them from infecting, unless your body is immunocompromised or you're able to evade your immune system. So microorganisms also use oxygen requirement. We shall come to that. Some also use nitric acid, sulfuric acid to produce energy. Some can live efficiently in chemical environments that are harmful to humans. And some of these are beneficial. So after the mining of minerals, these heavy metals, these harmful chemicals, could be fed on by microorganisms to make the environment clean or safe. So in the petroleum industries, they are making use of these microorganisms that are able to feed on toxic chemicals, pesticides, so that we can have a safe environment. So not all microorganisms are harmful. Just a few cause diseases. And the few that cause diseases, they are even suffering. There are many. So anywhere you find yourself, be conscious of your environment, because microorganisms are there. You cannot see them, so you cannot. So always protect yourself. And once we interact with animals, of the infections from them, they may be reservoirs that are not sick, and we will rather become the victims of such infections. We depend on these microorganisms system where they help in your digestive tract. They help with digestion, producing the right pH so that they can fight other infections or help you to digest your food. These microorganisms in your mouth, on the skin, are also so we need to be careful the environment we find ourselves in, and then how we deal with these microorganisms. That is why it's important that before and after work, you clean your surfaces, your benches, your floors, so that you are sure you have a safe environment for your clients and yourself. After work, you don't go without washing or cleaning your hands effectively before you meet your family. Otherwise, you may transfer microorganisms from the health facility to your house and infect your family members. So the conditions that they need, these microorganisms need to grow. It's not only bacteria, but I'm using bacteria as an example. You are a living organism, you need food. So microorganisms also need food. So nutrients to grow. But they don't have the digestive tract like you have. So they pick up simple molecules that will help them to survive. Then some like acidic environments, some like alkaline environments. So you should know where you pick the sample from. If it's in the environment and you want to culture, you should produce the same condition. If it's an alkaline environment, you have to produce the same environment in the lab. Otherwise, they will not grow. That is why some organisms can live in the mouth, but they cannot live in the gastrointestinal tract because they don't have the same pH. The normal flora in the gastrointestinal tract are not the same as the ones you have in the mouth. Temperature also affects how these microorganisms grow. For some of them, if you increase the temperature, they die. For some of them, if you reduce the temperature, they survive. So it affects how microorganisms grow. And if you want to kill them, you increase the temperature, you raise the temperature, and they don't survive, most of them, unless they are the hyperthermophiles. But the good thing is, those that survive very high temperatures do not cause diseases. So they have oxygen requirements. Some are aerobic, so they like environmental have oxygen. Some are anaerobic, environments that do not have oxygen to survive or for their respiratory activities. Water is needed for every organism to survive. So osmotic pressure is very, very important. If they lose a lot of water with time, they cannot survive, so they will die. That is why most of our foods are preserved by drying. The same way, if you take out water from the environment, most microorganisms will not survive. So talking about nutrients, they need carbon, they need nitrogen, they need inorganic salts and trace elements like copper, zinc. Some also need growth factors like the vitamins, the amino acids, the purines and the pyrimidines for DNA synthesis. They need water as well. So without these, microorganisms will not survive, even if you produce or provide the other conditions for them. So they use a variety of nutrients for energy. They need energy for their metabolic activities. Enzymes need energy to perform. So without the nutrients, the enzymes cannot perform the activities. DNA replication, reproduction will not go on because nutrients are lacking and the microorganism cannot survive. They also need these nutrients to build their structures. So the cell wall, the cell membrane, they need these nutrients to build them up. If there are destroyed parts of the cells, they have to quickly use some of these nutrients to build them. So about 95% of the dry weight of these microorganisms contain carbon, especially, and nitrogen, oxygen, and then hydrogen. So these are the major elements that form the bulk of the microorganisms. The other 5% are needed in small amounts or concentrations. So the calcium, the copper, and magnesium, the phosphorus. It's not that they are not important. They are very important, but they are needed in small quantities. For example, phosphorus is needed for DNA synthesis, but they are not needed in very high quantities or concentrations. So they can rely entirely on inorganic chemicals as their source of energy. And these are referred to as the autotrophs, self-feeding. See, they use these inorganic chemicals to produce energy. Others will also rely on the products from these autotrophs. So they require organic carbon sources of nutrients for their survival or as their source of energy. So these are called the heterotrophs or organotrophs. Then we have those that require high amount of carbon dioxide, unlike the others, before they can grow. So if you are culturing such organisms, you need to produce extra carbon dioxide. So you can take out the oxygen by putting a candle flaming in the incubator to take some of the oxygen out, and then you have carbon dioxide produced. Or you can produce commercial carbon dioxide so that you enhance the growth of these capnophiles, such as Campylobacter or Cobacter. The Campylobacter causes dysentery, can have diarrhea caused by Campylobacter. So pH or potential of hydrogen is very, very important for the survival of these microorganisms. If you want to culture microorganisms, you should know the pH at which they can grow best. And usually where you take the sample from or where the infection occurs gives you a clue of the pH to also provide when you want to grow them in the lab. So it's either acidic or alkaline, but some can hover around the neutral. So the Mycobacterium tuberculosis, the one that causes TB, prefers a slightly acidic environment, so pH of about 6.5 to 6.8. That is the optimum. If you lower the pH, they can survive, but the growth rate will reduce. So Vibrio cholerae, the one that causes cholera, also lies slightly alkaline condition. So if you culture it in an acidic medium, the growth rate will not be optimal. So based on the pH requirement, we can classify microorganisms as neutrophiles or neutrophilic, those that grow around the neutral, around 7, so 6 to 8. We can classify some as acidophiles or acidophilic, those that grow in acidic environments, those that grow in alkaline environments or basic environments, the alkalophiles or alkalophilic organisms, around 9 to 11. So we have the optimum that will allow or enhance their growth effectively. Away from the optimum, these are the peaks, these are the optimum. They can also grow, but the growth rate will reduce. So if it's an acidophile, if you move or reduce the acidic content, getting to the neutral, they will grow, but the growth rate reduces. If you bring them to the alkaline environment, then it's very toxic or detrimental for them, so they will not survive. So always, you should know the environment and then the pH of that environment where the organism was introduced. So for treatment, they can also produce the opposite of the pH that organism likes. So some drugs can produce the opposite pH so that the organism will not survive. So this is the optimum that enables them to grow for acidophiles, for neutrophiles, and then alkalophiles. So temperature is very important to maintain the three-dimensional shape of these macromolecules. So the DNA, the RNA, the proteins. Because the proteins are responsible for the activities within the cell. They control all activities because once the protein is synthesized, they have the genes expressed. Sorry. So once you increase the temperature, you destroy the hydrogen bonds that are within the DNA or some of these macromolecules, and it will halt or disrupt the activities within the cell. That is why you also always have to get the optimum temperature. So if you alter the pH, the temperature, sorry, so assuming the organism likes room temperature, and then you put it in the fridge, it will not die in the fridge, but the growth rate will reduce because the temperature has changed. Same applies to increasing the temperature, unless you provide the optimum for them. So the proteins can become denatured, destroyed, and the organism cannot function properly. If the temperature is too low, they can also break and then lose their cellular structures. So at very high temperatures, the lipids that protect the cell wall and the cell membrane, they can also become liquefied, and eventually the cell will lose its content and die. So you need to preserve the integrity of the cell so that you have the right conditions to make them survive. So treatment or control of microorganisms would mean that you have to also produce the opposite of the temperature that they require. That is why we use the autoclave, mostly in these facilities, in the hospitals. We use the hot air oven, the incinerator to produce very high temperatures and then get rid of these microorganisms. All right. So if the optimum temperature is 37 degrees Celsius and you bring the organism to a fridge temperature or room temperature, let's assume room temperature 22 degrees Celsius or 25 degrees Celsius, they will not grow very well. That is why in the lab, they put these samples in the incubator so that they can raise the temperature above the room temperature to achieve maximum growth. If you increase the temperature a bit, the organism will grow better than at room temperature, but it will not be as optimum as the 37, the one that cause most of these infections in humans. They prefer this 37 degrees Celsius because that is your body's normal temperature. So take note that once you put your food in the fridge, after preparing your stew, then you scoop some into bowls and you put in the fridge. If the stew is contaminated before you put in the fridge will not stop the organisms from multiplying. With time, you see that the stew or the soup is going bad. So always make sure what you put in is what you get. Don't expose your food to these microorganisms before you put them into the fridge. Always maintain a clean environment. So we have the minimum where the growth rate is reduced, then the optimum where the growth rate is highest, and then maximum where the growth rate is affected. So the maximum growth temperature increases, but the organism may not survive unless it is a hyperthermophile. So always look out for the optimum temperature. And fortunately, scientists have conducted a series of tests to know the optimum temperature for most of these infectious microorganisms. That is why you have to refer to the facility's standard operating procedure so that you can effectively culture or grow these microorganisms. So there are some infectious agents that prefer lower temperatures than their body temperature. So Treponema pallidum, the one that causes syphilis, or Mycobacterium leprae, the one that causes leprosy. Another example is the Mycobacterium leprae. This one is difficult to culture on artificial media. So this is an exception to Koch's postulate. So we have growth requirements for these microorganisms. Those that grow at very low temperatures are called the psychrophiles. So they can live in the fridge, in the freezer. That is their optimum. But the good thing is they don't cause infections. Most of them are less harmful or non-harmful. Then we have the mesophiles. These are the ones that cause infections. So look at the temperature range, around 20 to 40 degrees Celsius. So the optimum is your body temperature, 37. That is why most infections are coming from these mesophiles or mesophilic bacteria or microorganisms. Then thermophiles, they can withstand some degree of temperature higher than 40 degrees Celsius. And that is the optimum for them. So for such organisms, if you perform pasteurization at 65 degrees Celsius, they are not affected. So if you want to warm the milk of your baby around 50 degrees, 60 degrees Celsius, the organisms that are able to cause infections, the mesophiles, they will not survive because it's out of their range for survival. So pasteurization will work against those mesophiles, but the thermophiles will grow effectively in your product, your milk or your food product that you want to pasteurize. But most of these thermophiles do not cause infections. That is why our babies are safe. They have the hyperthermophiles, and these are able to withstand the boiling point. So you boil your food, they still survive. But the good thing is they are not causing infections, diseases. So if you want to culture, you know the organism and then the suitable temperature. There are other fields of microbiology that make use of different temperature ranges, depending on the field of microbiology. So the environmental microbiologists may be interested in the hyperthermophiles or the thermophiles. But you are the medical workers, so you will be dealing with organisms that are mesophiles. So to control these microorganisms from growing, you can reduce the temperature or increase the temperature. If you reduce the temperature to about 10 or 8 or 4 degrees, which is about the temperature of the fridge, they will not die. They will survive, but the growth rate will be reduced. That is why we encourage heating to control these microbial growths, or you can use chemicals to control microbial growths. So oxygen requirements, some like oxygen, some do not like oxygen. So we have the aerobic and anaerobic microorganisms. So mind you, bacteria is a group of microorganisms, not the general term for all microorganisms. So oxygen is essential for obligate aerobes. So aerobic aerobes, obligate, oxygen only. When I say oxygen only, I mean they need oxygen for their respiration. In the absence of oxygen, they will die. Then we have obligate anaerobes, the opposite of the aerobes. So they don't need oxygen, respiration by the absence of oxygen. So based on this, you can conduct a simple experiment in a test tube with media. So the media has the nutrients that they need for their growth. So if you put organisms in these 5 test tubes to see the oxygen requirement, you are likely to have these 5 groups of organisms based on their oxygen requirement. So we can have the obligate aerobic bacteria. They like oxygen at all costs. So they will settle at the top because this is not a closed test tube. So they will come to the top to have access to atmospheric oxygen. So you see them settling at the top of this test tube. They are the obligate aerobes. They need oxygen for respiration. If they move down here, they will die. Then we have the obligate anaerobes, the opposite of this one. So opposite of the top is bottom. So they will move to the bottom because they are anaerobes. They don't need oxygen at all. It is toxic for them. Then we have the facultative anaerobes. facultative, they can switch. So look at those ones where they are mostly at the top. So facultative anaerobes like or prefer oxygen for respiration, but in the absence of oxygen, they can also respire and then survive. That is why some are even here, but they prefer oxygen. They are not affected by the absence of oxygen because they are facultative. They can switch and use both means of respiration, aerobic and anaerobic. So they are the facultative anaerobes. But because they have the access to oxygen, most of them will settle at the top. But moving to the bottom, they are not affected because they can survive in the absence of oxygen for other means of respiration, which is the anaerobic. And we have the microaerophilic or microaerophiles. Looking at this one and then the first one, where they settle at the very top. This one, they move to the upper part, but not at the very top because they need oxygen in very small quantities. They are microaerophilic. Micro means small. They need oxygen in small quantities. That is why they move to the upper part, but not the very top like the obligate aerobes. Then we have the aerotolerant. They are not affected by oxygen, so they tolerate any condition. They tolerate any condition, and there's a slight difference between the facultative anaerobe and then the aerotolerant. Facultative anaerobe, they prefer oxygen, but absence of oxygen, they can switch to anaerobic respiration. Aerotolerant anaerobes, they are able to live in the presence of oxygen, but they use the anaerobic respiration, unlike the obligate anaerobes, where they will move to the base. I hope it's clear. So if
Please,
yes, Esther, let's hear you.
Hello, sir. You
Yes,
said
Esther.
the facultative, they like oxygen, but in the absence of it, they use the anaerobic.
Yes.
So, sir, please, with the facultative and the micro aerosols, when you arrange it, like in terms of the arrangements, which one will be top and which one will be down?
How? So we have A, B, C, D, E. The A, B, C, D, E does not mean 1, 2, 3, 4. It doesn't correspond to what is on the right side.
No, yes, I understand, but I'm trying to say that it says the facultative, they need the oxygen, but in the absence of it, they can survive.
Yeah, so let me tell you the trick. It's facultative anaerobe. You get it? So they are aerobic, but they can switch to anaerobic state of respiration. So once you see the facultative anaerobe, it means that they prefer the opposite, which is the aerobic. But they can switch to the anaerobic respiration when oxygen is absent. That's why you call them facultative. Faculty, they can switch. They have options. Then you said the microaerophilic, right?
Yes, please.
Microaerophilic, they need oxygen for respiration, just like the obligate aerobes. So for the microaerophilic, if you bring them to the bottom where oxygen is absent, they will die. But the oxygen that they need is in very small concentration. That is why they don't settle at the very top, like here, but they move a little below the upper part because they need oxygen, but small form, dissolved form. They will not come to the very top to have direct exposure to the atmospheric oxygen. That is a micro. They need oxygen at all cost, but in small quantity, which is different from the obligate. At all costs, they need oxygen. If you bring them to an environment where oxygen is lacking, they will not survive. Esther, is it clear, please?
Professor, please, from your explanation, if there is the exposure of oxygen, facultative anaerobes, like they will gravitate towards it more as compared to the micro aerosols.
Exactly.
Okay, thank you.
Because they prefer oxygen. But if you take oxygen from them, they are not bothered. They can switch to anaerobic respiration because they are facultative. They can switch. Is it clear?
Yes, please. Thank you.
Yes,
Yes, who is lost? So that we can find you.
self the one. My
All right. Where are you lost?
this particular slide, I'm not okay with it.
Okay, so microorganisms live in environments that could be very rich in oxygen. Some live in environments that are without oxygen. So assuming in your body, some organisms settle in the lungs where you have oxygen abundant. If you're exposed to obligate anaerobic bacteria, it will not survive in the lungs because it is not able to survive where oxygen is present. So when we talk of obligate, that means at all cost, it needs oxygen. Obligate aerob, it needs oxygen. Obligate anaerob, at all cost, it needs absence of oxygen. So the first one is an obligate aerob. That is why you see them coming to the top for oxygen, the very top. The obligate anaerobes will move to the bottom where there is no oxygen. So that's the difference between the obligate aerobes or obligate aerobic organisms and then obligate anaerobic organisms. Then this is where most people get confused. Facultative anaerobe or facultative anaerobic microorganism. So the emphasis is on anaerobe. That means they prefer oxygen. Facultative anaerobe, they prefer oxygen. So if you put them in an environment where oxygen is present and then on the other side oxygen is lacking, most of them will move to where oxygen is present because they are facultative anaerobes. But those that move to the opposite side where oxygen is lacking, they will not die because they can also switch to anaerobic respiration. That is why you see some moving to the bottom. So once they move away from the top to the bottom, they are undergoing anaerobic respiration. That's why they are facultative anaerobe. Then we have the aero, please mute. We have the aero tolerant. They tolerate oxygen, but they are anaerobic. So they will rely solely on anaerobic respiration. But presence of oxygen does not bother them. They are not bothered by the presence of oxygen because they will not need oxygen to do anything. They will use other means to undergo anaerobic respiration. The microaerophilic, they need oxygen but they will need it in small concentrations, so they will come to the upper part, but below the very top. I hope it's clear. Yes, Damata, your hand is up.
Yes. Please, I want to get the understanding when you talk of anaerob, meaning no oxygen, or please.
Yes, so aerobic
Uh-huh.
means oxygen, anaerob means no oxygen.
Mm-hmm.
But when we say facultative anaerob, that is where people get confused. Facultative anaerob is the type that needs oxygen. They prefer oxygen.
Yeah,
But
I'm a bit
if
confused there because I'm thinking, why is it not facultative,
facultative
rather
aerob,
facultative anaerob? If you
yes.
are saying they
So
love oxygen, but they can
yes.
switch when there
So
is no oxygen.
let me use this simple experiment example.
Okay.
You are a midwife,
Yes, please.
and you have nurses in the facility.
Yes,
When
please.
you come to work, your duty is to perform that of what? The midwife.
Yes,
So
please.
let's say oxygen is the duty of the midwife.
Okay.
So when you go to work, you use oxygen, so you're a midwife.
Okay.
When you exhaust your work and they need somebody to help with the nursing, you cannot say I cannot do it. You can survive among the nurses.
Okay.
Yes, but you need oxygen. If oxygen is not there, you can still undergo anaerobic respiration. But you prefer oxygen if it's there. So once we use the facultative, then it goes with the opposite of what it can survive on in the absence of what it prefers. If you say it is facultative aerob, that means at all cost, it will prefer anaerobic condition. But if the organism finds itself in an aerobic condition, it will be able to undergo aerobic respiration. But we don't have such organisms. Most of them are the aerobic that can undergo the anaerobic. So typical example is you culture them and oxygen is depleting. If oxygen is depleting, they can undergo the anaerobic respiration. So they are facultative anaerobes.
Okay.
Somebody says you will become a facultative nurse. So that's a facultative anaerobe.
Okay,
So
thank
the
you.
problem says, so the facultative anaerobic, they can live both with or without oxygen, but they are good to go with oxygen, the facultative anaerobe. If you provide these options, they will go to the aerobic part. But those that go to anaerobic part, they will survive. It's just like when your postings came, you prefer your hometown. So the hometown is the aerobic condition, but you are facultative anaerobe. You can survive outside the hometown. All right. Thank you. So there are different types of organisms. So all these are bacteria anyway. We shall come to the other groups of microorganisms. So I'm using bacteria as an example. That are aerobes. So Pseudomonas aeruginosa, Mycobacterium tuberculosis, Bacillus subtilis. Most of these are the ones that cause the upper respiratory. because oxygen is favorable to them. Then Clostridium, the bacteroids, they are anaerobes. So when you want to culture them, you have to remove the oxygen concentration or content from that. Then E. coli, Staph aureus, they don't mind switching between aerobic and anaerobic conditions. Then aero-tolerant anaerobes, they are not bothered by the presence of oxygen, but they will go in for anaerobic respiration. So Streptococcus, Campylobacter,
Sir,
Lactobacilli.
please, though.
Yes, please.
With the anaerobes,
Mhm.
if you want to disable them, do you expose them to oxygen or you deprive them of oxygen? I'm getting confused here.
The anaerobes, if you deprive them of oxygen, that is hooray for them because they don't like oxygen.
Okay.
So for
So you
such organisms,
expose them,
yes, you expose them to oxygen.
but you said otherwise. That was why I was asking.
Unless they are obligate anaerobes. So you get the difference. If they are obligate, that means oxygen is toxic for them. So we have
Okay.
obligate aerobes. For obligate aerobes, oxygen is what they need. They don't need carbon dioxide. They don't need environment that is devoid of oxygen. If you put them in an environment that has no oxygen, they will die. Then the obligate anaerobes, they want or they need only environment that is absent in oxygen. So they will move to the bottom, sorry. But it is facultative. That is where they can undergo both aerobic and anaerobic. So facultative anaerobe, they will prefer oxygen. That is why they are coming to the top. But they can switch to anaerobic respiration when oxygen is absent. So I hope it's clear. The one who asked the question.
Yes, sir. Yes,
Yeah,
sir. Thank you.
the microaerophilic.
Hello, sir. Hello, sir.
Yes,
Please,
please.
good afternoon. This is Rose.
All right. Let's hear you.
Please, I'm having some example. I want to share my example and see whether I'm correct or not. The the first one, that one you said the person lacks oxygen. So if you take a fowl like this, and then you put a fowl in the water, that fowl may die.
That is
But if
obligate aerobe.
there is oxygen, the fowl will survive. Then the second one, you take a frog or a fish. You know, fish, they like water.
Yes.
So maybe the fish, it doesn't mind. You put the fish inside water, whether there's no oxygen, but the fish can survive. But if you put a fowl in the water, the fowl will die. So the fish will be the second one. Obligated, or is it what? Like the names, you know.
Anaerobe.
But I'm getting obligated. Then the third one, the facultative anaerobes. That one is both. You
So
know,
like
frog
the frog.
like
Yes.
frog like water, and the same time too, frog can also survive at the island, like the land. The frog can move in the land and then the same time in the water. So if
Or the
you cook a
crocodile.
frog yes. So I don't know whether you can explain more so that we'll get it.
So
That's
let me
where
use
I'm getting at.
let me use the frog as an example.
Yes.
You see the frog on land where you have oxygen. So it is as an example. I'm not saying that the frog is facultative anaerobe. So the frog, if we were to look at microorganisms, the frog would be the facultative anaerobe because it prefers the environment that has oxygen.
Yeah.
But if you put the frog in water, it can switch to the anaerobic respiration where you don't have oxygen. So the frog can be an analogy of the facultative anaerobe.
Okay.
Is it clear, please?
Yes, please.
Right.
Thank you.
Thank you. So we have about 5 minutes. If it ends, we will continue from where we will end, and then if you have questions, we can tackle those before we start the next lecture. So microbes and water. You know, every organism needs water. If you put an organism in a condition that has more water outside than what it has inside, so that environment is a hypotonic. So hypotonic means it has less solute, so more water in the environment than what is inside the cell. There will be movement of water from the environment that has high concentration of the water to the inside. So the cell begins to expand. So bacterial cells, they have the cell wall, the cell membrane. The cell membrane begins to expand until it reaches the elastic limit. And if water continues to move inside the cell, then the cell will burst. So water can affect how microorganisms also survive. If you do the opposite, where you put the organism in a hypertonic environment, that means the environment has less water than what the cell has or what the organism has. That means the organism is going to lose a lot of water to the environment because the environment is hypertonic to the cell. So if you put salt in your tilapia and there is bacteria in the tilapia, the environment has little water compared to the cell. So the cells within the organism will lose water towards the salt, the environment, which is hypertonic. So with time, the cell begins to shrink and the water content reduces and the cell cannot function effectively. That is why microorganisms cannot survive in gari, in your tilapia, the dried one, the dried fish. But if you put the fish that is moist there, within some few hours, it begins to go bad. Because these are the requirements for growth. Every organism needs water. So if it loses a lot of water, it becomes plasmolized by the high concentration of the solutes that are on the outside of the external. Water moves out and then it loses water. It loses water, so it becomes desiccated, undergoes hypoosmotic shock. But there are some organisms that are very smart. When they realize that the water content is getting depleted, they undergo endospore formation. So they form a tall coat around them. And they come to the resting stage where they don't reproduce, but they are not dying until conditions become favorable, and then they come back to the vegetative reproductive form. So some can form endospores and then cysts. We shall look at bacteria and then the others that form the cysts when you look at the classification of these groups of microorganisms. So we have environments that these organisms can live. Microorganisms are everywhere, but you will not find the same organism everywhere because of their preference. So we have the halophilic that live in high salt concentration, like the seawater. Then obligate means at all costs they need high salt content. Extreme, that means very high salt concentration. Facultative, they can grow with or without high salt concentration. They can switch between the levels. So they can live in seawater, they can also live in fresh water. They can live in the human body also, where you have the low salt concentration in the blood. the human body provides all these conditions that we have talked about: nutrients, pH, temperature, water, for their survival. So your body is an excellent incubator. That is why we are prone to infections. So we need to prevent these organisms from having access to the human body. Otherwise, they will multiply because the conditions there are very favorable. So that's the end of today's lecture, Unit 1. History, habitat, and then conditions that are necessary for microbial growth. Any question before the system closes us down?
Sir, please, can we say that the facultative can be very difficult to be getting rid of? Since they can survive
yes, so
with or without oxygen.
Yes, so mostly those ones, they can switch to the aerobic and then anaerobic. So most of them can cause infections in several parts of the body. That's why you can have infection by E. coli in the urinary tract. You can have them in the gastrointestinal tract. You can have them in the blood because they can survive in aerobic and anaerobic conditions. But some organisms, you will not find them. For example, Mycobacterium tuberculosis, you will not find them in urinary tract infections because the pH and then the oxygen concentration will affect them. So those facultative anaerobes are the ones that are common when it comes to infectious diseases. The common ones that we have, but temperature, of course, affects these organisms as well. Yeah, Esther. Esther,
Sir,
please.
Sir, please, with the osmotic pressure, I want to know if I understood what she said. You said that if there is more water outside the cell, it goes into the cell, then it's
hypertonic. Yeah, so if you have more water outside, that is hypertonic environment. So it is hypertonic to the hypotonic. Hypo means less. So it has less. Tonic means solute. It has less solute than water outside. So if it's hypertonic environment, that means you have more water than what you have inside the cell. And water will move towards the inside.
Okay.
By passive transport. It's the river that needs energy. If it's active, that means it needs energy. You are bringing it against the concentration gradient. But for water to move from the outside to the inside, it's passive. It doesn't require any energy. That's why it will flow from the outside to the inside, and then with time, the cell expands, and then will burst.
Okay.
I hope it's clear.
Yes, please.
Someone has a question.
Yes, sir.
Oh, if you don't understand, please feel free. That's why we are here.
Say. Hello, say.
Yes, please.
Please, with the erotolerant anaerobes. That one, I didn't get the explanation very well.
So, aerotolerant anaerobes, they tolerate oxygen, but they will not use oxygen for respiration. Please mute.
Who is that?